Hey all, so today's video, we're going to be looking at C4.1, which is talking about populations and communities on page 501 to 528. This is looking at the theme of interaction and interdependence because organisms in ecosystems tend to interact with each other and then also rely on each other in certain ways. So we want to be thinking about how do interactions between organisms regulate the size of a population within the community, and then what interactions within a community make its populations interdependent. So firstly, we need to have a definition of a species. A morphological species concept from the 1850s would have been that they have similar internal and external form and structure, but that doesn't align with natural selection. And the reason for that is that different organisms can have different internal or external structure or form, such as octopuses having the same eye structure as humans, but not be the same species at all. So now we have a biological species concept, which is groups of organisms that can interbreed to produce fertile offspring. And no fertile offsprings means that they're not the same species. They have diverged sufficiently to have been considered as speciated, which means they've become their own separate species. So for horses and donkeys, they speciated about 4.5 million years ago, whereas polar bears and grizzly bears speciated about 500,000 years ago. So there's a few different factors that are challenging when determining whether or not a species is said to have speciated. And so obviously, if they can interbreed, so for example, tigons, which are a male tiger and a female lion, the females are sometimes fertile. So would you then say that lions and tigers have speciated? Yes, they have. And a reason for that is because they wouldn't naturally interbreed in the wild. And so grizzly bears and polar bears live in different areas. They are culturally, with mating rituals or behaviors, probably different. They probably smell quite different. They're unlikely to mate in the wild. Does that mean that they couldn't mate in captivity? Potentially they could, and they could potentially produce some fertile offspring, but they would be somewhat limited. Variation is happening naturally in an ecosystem, in a species, and it's required for natural selection to happen. We're going to look at natural selection in a lot more detail later on, but natural selection is where the environment is selecting traits that are advantageous to that organism in that environment. And so the way that that selection is happening is the organism that has those traits that are advantageous survives longer, gets food better, reproduces more, and passes on their genetics to their offspring, and their genetics may have that trait. If you didn't have variation happening, then a population could be very successful within a certain environment. But if that environment changed, then all of those organisms could die and the species could go extinct. And so variation is critical to a changing environmental situation. If we were looking at the least genetic diversity within a species, that would be monozygotic twins. So those are identical twins. They come from the same egg and sperm being fertilized and then split into two zygotes. So as a byproduct, they are genetically identical. So if we're talking about populations, what is a population? This is a group of organisms that belong to the same species and that live in the same area at the same time. And so you can see this population of deer here, and they can be used to study genetic diversity and evolution. So how has this population of deer changed from different other populations of deer that might be geographically isolated from these deer? And you can also look at, using DNA testing, how similar are these deer here to each other. Hybridization is when species migrates into a new area and they are closely related to the resident population. So they haven't fully speciated, perhaps they lived apart, or typically they've lived apart geographically, and so they're not going to be able to interbreed at that time. but they will slowly change over thousands of years to become separate species. But if they're then reintroduced, they could breed with the local population. And as I said before, with polar bears and grizzly bears, scientists will say that species are separate if they are unlikely to breed in the wild. So species populations are going to be needing to get nutrition and some vocabulary that we need to know for this. Herbivores eat plants, carnivores eat meat, omnivores eat plants and meat. Autotrophs create their own food from sunlight, so plants, and they're going to absorb nutrients from the soil in the abiotic environment. Heterotrophs eat things to get energy. So humans are heterotrophic omnivores. Saprotrophs, however, are going to be heterotrophs that externally digest. And so mushrooms excrete enzymes into the dead material that they are going to digest, and then they suck up the juices from that digestion process. So they're still getting energy from food, but that food is external to their body, and they're digesting it externally. And then detritivores are going to digest waste for food. And so an example of this would be a worm that eats, let's say, fecal matter, eats soil and digests the organic components out of the soil and then poops out the rest, but they're still digesting internally. So if we're looking at populations, we would need to potentially measure the size of a population. A carrying capacity is the population and environment can support based on the resources available. So water, light, soil, space, food, oxygen, how many organisms can live in this area. And when we are measuring a population size, we want to be doing random sampling, which is going to allow us to have an equal chance of selecting whichever area we're going to be measuring. And so if we're looking at quadrat sampling, this is really good for sessile, meaning non-moving organisms. But like plants, for example, you could imagine even butterflies or deer or birds or something, they're not going to sit there while you go around and count them. So as a byproduct of that, a different measuring strategy is going to need for motile organisms. But if we're using quadrat sampling for sessile organisms, then we would want to use a random number generator to know which of these squares that we are going to choose. So it just minimizes sampling error and our own bias in which areas we are choosing. If we are looking at motile organisms, the way that we can measure them is to use a capture, mark, release, and recapture strategy. So for example, if you were looking at raccoon population in a park or a forest, you would capture some raccoons, tag them all, record the number that you had captured, and then you would release them and then recatch. And based on how many were tagged initially in that recaptured population, you can use the following Lincoln index calculation to determine what is the likely population of your raccoons. So you'd want to do this a number of times. And the reason for that is so that you could figure out a standard deviation value. Obviously, as we've seen in many labs, standard deviation, high value, if for standard deviation means there's a lot of variability from the mean, whereas a low standard deviation means that the results that you're getting are quite close to the mean. And so optimally, you would want values that are close to the mean, unless there is a high amount of variability in your results, in which case that high standard deviation is telling you that there is a wide range of results in that population. All right, so populations change over time, and they're going to remain stable due to negative feedback loops. And we've already talked about negative feedback loops as it relates to homeostasis. It's quite similar when it's related to populations. And so here we might be looking at, here's the carrying capacity, and you can see that the population is stabilizing around the carrying capacity. So what might be happening there is as the population grows, not enough food is available, and so it dips. And then as it dips, it becomes more food available, so then it grows again. But there's density dependent and density independent factors that can affect a population as it grows. Density dependent factors will affect a population as it grows. Density independent will not. And density dependent factors are going to result in negative feedback loops. And so density dependent factors are that as a population gets bigger, then the population is going to decrease due to some factor related to how many organisms are there. So it could be related to competition. They're just competing for too much food. Could be disease. It could be parasites increase when the population increases. It could be that there's some kind of infestation that occurs. It could be that predators increase when the population of prey increase. And so these are all going to result in a decrease. So you have an overshoot from the carrying capacity, and maybe due to disease, the population drops. In the case of density independent factors, the population, it doesn't matter how big it is, it's going to drop because of some outside factor. So that could be something like a forest fire. And or it could be an introduction of a disease, but it was unrelated to the population of that organism. So the population would then drop, and then it would slowly crawl back up. In the case of predators and prey, you can see here in this rabbit fox diagram, there's more of a boom and bust cycle with the rabbits and the foxes. And there's a bit of a time lag going on there. So what's happening is as the population of rabbits increases, then the foxes have more food, they can have bigger litters of puppies. And so as a byproduct, they will end up the rabbit population will really drop, and then a little bit later, the fox population will rise once the rabbit population is high. So this is still hovering around a carrying capacity. It's just that the fluctuations are larger. And when we're looking at this curve here, initially the population increases almost exponentially. And this is called a J curve. And J curves are then going to stabilize around a carrying capacity in many cases. And so J curves usually will turn into an S curve over time. Reproduction is a positive feedback. So that's this J curve initial growth process. So meaning once there are more organisms, then more organisms will have more babies. And that's going to lead to growth in a population. In the case of the Eurasian colored dove, it was introduced from Asia to Europe, but didn't have any significant predators in the European environment. And so as a result, its population grew and grew and grew, but then it was controlled by density dependent factors. As the population got too big, they were then competing for space. They were competing for food, and so that kept their population under control. Nextly, we're going to be talking about community. And so we've talked about population, but a community is all the different populations living in the same area at the same time. So a coral reef would be a good example of a community. All the fish, the coral, the turtles, all of those organisms together are a community. And it's all the biotic factors there. And these can be used to study biodiversity. So different types of organisms that live in an area and also their genetics. Interactions between populations in these communities may harm or help other communities, other populations. So we have intra-specific interactions. That means that individuals of the same species are interacting together. And so that could lead to competition or cooperation. In the case of competition, we might be competing for mates. We might be competing for space, water, breeding sites, light, food. In the case of cooperation of the same species, it could be like wolf packs working together, penguins huddling together to keep warm, humans helping each other. So intra-specific interactions, same species, and they are working together or opposite to each other. And it really depends on the situation. Intra-specific looking at this next part, an ecosystem is going to include all the living organisms in a particular area, as well as that physical environment. And so that's going to be the biotic and abiotic factors in an area, which is a bit different than a community, which was only looking at the biotic factors. So then looking at interspecific, this is between species. So this could be herbivory, a deer eats a plant, but the plant might not die. Predation, so foxes eating rabbits. Mutualism, where they're both benefiting. So this could be something like bees and flowers. It could be something like root nodules that contain bacteria in legume plants like peas and beans. And the bacteria help give the beans nitrogen resources, and the beans give the bacteria sugars. So lots of different types of mutualistic examples. Parasitism, where one benefits and the other is harmed. So this could be like ticks or tapeworms, for example. Pathogenesis, I can't believe I can't say this. Pathogenicity, there we go, where one is causing a disease. And so this could be, again, from ticks, it could be from mosquitoes, it could be from bacteria or amoebas, for example. Resource competition. So this could be, this is between often endemic and invasive species in an interspecific interaction, but it doesn't have to be endemic versus invasive. It could just be two endemic species competing for food. The word endemic means that they live in that space naturally, whereas invasive means they've been introduced. And often invasive species introduction can be problematic because those invasive species don't have natural predators in that environment, that new environment. And so they can outcompete the endemic species that do have predators. And that could lead to competitive exclusion, meaning the invasive species takes over. So an example of this could be the lionfish in Florida. It was introduced from Asia, and it has these spines on its body that are venomous. And so it has no natural predators in a Floridian environment. And so it's competing for food with other organisms and outcompeting them. And it's also true of the cane toad that was introduced to Australia from Africa. And they get really, really big. And so they're so big that they will eat up everything in an aquatic ecosystem, including the other frogs. Sometimes they will even eat birds, which is just insane to me. And then we can measure interspecific interspecific interactions using something called a chi-squared test. So are these organisms interacting, or do we find one and the other together? And so that could look like in a forest, like lingonberries and blueberries. Do they grow together or not? And so if not, then we might draw some conclusions. Why not? But we would use a chi-squared test, and we'll have a separate video about this as a statistical test to determine if there's any statistical significance of these two species growing together. So we can have top-down and bottom-up population control. And so what that's going to mean is that top-down control, as we increase the number of predators at the top, it's going to control the number of prey at lower levels. And so this can be positive for an ecosystem because it's going to limit the number of herbivores, for example, which would then allow vegetation to grow more effectively. And so we saw this when we looked at the wolf video from last year, how wolves control rivers. And so wolves were introduced, and then they controlled the deer population, which allowed a wider variety of species to move into that area because they had a wider range of access to vegetative locations. And then bottom-up control is where population of producers may be limited by nutrients in soil or water. So how many trees there are, how many plants there are. And so higher trophic levels are going to be limited by how much vegetation there is. And so we don't see big predators in a desert environment because there's just not enough plants to support that middle layer, that herbivore layer. And so trophic level is what I mean when I say layer. And so there just wouldn't be enough food for a big predator to live in the desert. So an example of this, certain ponds in the UK, there's bottom up and top down going on here. So if the water is shallow, then the plants can root. So this would be bottom up. And that gives shelter to fish, and algae doesn't grow very much because fish are the top of the food chain and they are eating that algae. In deeper water, there's not enough sunlight. The plants can't root and the fish have no shelter. So then the fish die and then the algae grows too much. So in this case, the algae is what needs to be controlled by the fish. That would be more of a bottom-up situation. And the rooted plants are more of a bottom-up control. So when we're thinking about populations and communities, we may think about antibiotics, and antibiotics are secreted by funguses to combat bacterial growth near them, because funguses are actually getting nutrition from the same resources as bacteria will, so like dead stuff. And so they're producing these secondary metabolites to limit the growth of bacteria. And so that would be how antibiotics are formed, and the way that scientists determine or create antibiotics. It's to look at a wide range of different funguses, what bacteria they control. And that was how the first antibiotic, penicillin, was first discovered, because blue-green bread mold was limiting the growth of bacteria populations on sample plates. Allopathic agents are when a plant secretes chemicals that are going to limit the growth of competing plants nearby. So they don't want other plants stealing their resources. And so they're going to produce chemicals that are going to limit the growth of this other plant. And so an example of an allopathic agent would be that black walnuts release this chemical juglone to prevent other plants from growing nearby. And so if you were to plant a black walnut tree in your yard, you would have to anticipate that nothing really is going to grow around it. All right, so to summarize, populations can be measured using Lincoln Index, quadrat sampling, or chi-squared. Populations fluctuate based on density dependent or independent factors and can be controlled top up or top down. Sorry, top down or bottom up. Predator-prey interactions, pathogenicity, parasitism, mutualism, resource competition are all interspecific, meaning that that's happening between two different species. Intraspecific interactions can be cooperative or competitive. And funguses produce antibiotics, whereas other plants are going to produce allopathic chemicals to reduce competition between nearby other plants. All right, thanks for watching. Sorry, this one was a little bit long.